I would like to thank the organizers for giving me the opportunity to share our work to develop this limbo stem cell therapy. My presentation will be divided into 4 parts: background, then development of the therapeutic candidate, followed by development of the outcome measure, and finally, the phase 1 clinical trial design. Cornea is the transparent part of the eye, is lined by epithelium, and this epithelium is maintained by a group of adult stem cells that reside in the limbus, which is junction between the cornea and conjunctiva. In the basal layer of the limbal epithelium, and those layer harbors these group of stem cells. When they are deficient or lack of functional stem cells, that lead to a disease entity called limbo stem cell deficiency. The presentation of this disease varies based on the degree of the limbo stem cell deficiency. In partial disease, only a sector of the cornea and limbus is affected. In total limbo stem cell deficiency, the entire cornea is affected. Cornea transplant does not treat limbo stem cell deficiency. In eyes with normal stem cell function, the graft can remain clear and survive for a longer time. But in eyes with limbo stem cell deficiency, the graft usually fails within a couple years. The cause of limbo stem cell deficiency could be due to genetic disorders or due to injury. And any injuries to the limbus can lead to stem cell deficiency, which include chemical injury, contact lens wear, or chronic inflammation. It's not until recently that the first global consensus on this disease entity was developed. There are 4 take-home messages. The first is that the hallmark of the disease is the cornea epithelium is replaced by the conjunctiva epithelium. The second is that a diagnostic test is recommended in addition to clinical exam. Third, autologous stem cell therapy is preferred over allogeneic therapy. And lastly, limbo stem cell treatment is preferred over keratoparesis, which is artificial cornea. We also conducted a meta-analysis on the outcomes of limbo stem cell transplant. We found that autologous limbo stem cell transplant had a higher success rate and lower complication rate than did allogeneic transplant. And the long-term success rate is below 76%. The criteria of diagnosing and staging limbo stem cell deficiency and the outcome measure of any treatment vary greatly among all studies. And we concluded that randomized controlled clinical trials are necessary to compare the efficacy among different therapies. The first approved stem cell therapy was first reported back in 1997. In this first trial of therapy, a small biopsy was taken from the donor eye, which is the healthy eye, and these stem cells were then expanded on the dish and then transplanted onto the other eye, which is diseased eye, and the ocular surface was restored. The therapy was approved by the European Medicine Agency called HoloClear. There are several disadvantages of this therapy. First, it requires animal products. Second, it treats mostly unilateral disease. And it lacks a standardized outcome measure. And the success rate, again, was limited to 76% with repeated treatment. So looking forward, we think that the therapy should be safe, which means that it should eliminate animal products to avoid cross-contamination. It should be efficient, which means that the expansion of the stem cell population in culture should allow for a higher success rate. And third, it should be very effective. That means a higher long-term survival of these transplanted cells. So in the next session, we want to share with you how we develop a new therapy using cultivated autologous limbo stem cells. We focus on the manufacturing process of these autologous cultivated limbo stem cells. We want it to be feasible, efficient, and GMP compliant and xeno-free. To achieve that, we optimize the culturing process by improving the following categories. First is the starting material, which means that when the tissue is biopsy, how do we culture the stem cells from the biopsy? Secondly, are there supporting cells, feeder cells, any, that will increase the efficiency of the expansion of the stem cell population? And third, how can we ensure the culture substrate or the transplant substrate safe and effective? And the next, the culture media, how can we make it xenobiotic free? And lastly is the way that the cells are cultured and how can we recreate the in vivo limbo stem cell niche. So after comparing more than 70 different culture methods, and we finally develop a xenobiotic-free and feeder-free culture system by preserving the native stem cell niche during culture and then replacing the fetal bovine serum with human serum, and also removal of the DMSO and chlorella toxin during this process. Next, to comply with the regulatory requirement, we also develop IPCs in process controls and also release criteria so that we ensure the quality of the cultivated limbo stem cells. Next, I'm going to talk about the development of standardized clinical outcome measures. Clinical presentations might not reflect the actual stem cell function, and a quantitative staging system is still lacking and needs to be developed to quantify in vivo limbo stem cell function and outcomes of the stem cell treatment. We utilize a very powerful in vivo imaging, in vivo confocal microscopy. It can image all layers of the cornea, different layers of the epithelium, and the junction of the conjunctiva and cornea, which is the limbus. You first set out to look into the microstructural changes of the epithelium by imaging the central cornea and 4 regions of the limbus. Even in earlier stage, the epithelial cells become larger and the nuclei become more prominent. These changes, we call it metaplastic change become more apparent in the intermediate stage, and the cells with normal looking cornea phenotype are missing in the eyes with late stage. A similar picture is seen in the limbo regions. Often we see an influx of inflammatory cells. At the same time, we also develop a quantitative clinical scoring system that takes into consideration of the extent of the limbo and corneal involvement, as well as the central involvement. And each eye will be giving a number to quantify the degree of the clinical degree. We look into 24 in vivo parameters of eyes with limbo stem cell deficiency, basal cell density, epithelial thickness in 5 locations of the ocular surface, central cornea, and 4 regions of the limbus. Cell morphology, central corneal subbasal nerve density, and nerve branch scores. The basal cell density decrease in the eyes with limbo stem cell deficiency in both the central cornea and the limbus. The degrees of decrease correlate with the degree of the severity of the disease. Epithelial thinning also was detected in eyes with the stem cell deficiency. In normal eyes, the epithelium is visualized by another in vivo imaging modality, anterior segment OCT, optical coherence tomography. In eyes with stem cell deficiency, the layer of this epithelium becomes thinner and often absent in advanced stage of lymphostem cell deficiency. And the decline of subbasal nerve density was also observed. Again, the degree of decline has positive correlation with the degree of the deficiency. A molecular diagnostic test also is essential to confirm the diagnosis of lymphostem cell deficiency. We found that keratin 13 is a specific marker of the conjunctival epithelium. By sampling the ocular surface on the cornea, on the conjunctiva, and the cells can be subject to immunohistochemistry staining, and K13 positive cells on the cornea could signify lymphostem cell deficiency. So, based on our finding, we include clinical scores, basal cell density, subbasal nerve density, epithelial thickness of the central cornea, and molecular diagnostic test using keratin 13 as a marker could be used to diagnose and stage lymphostem cell deficiency. And this criteria will be used as outcome measure of our ongoing lymphostem cell trial. In this last session, I will introduce you to our phase 1 clinical trial. The purpose of this clinical study is to investigate the safety and feasibility, hopefully part of the also efficacy, of our cultivated autologous lymphostem cells for the treatment of lymphostem cell deficiency. We also want to validate the lymphostem cell deficiency diagnostic criteria and quantitative staging system. This is a randomized controlled phase 1 clinical trial at a single center. 20 subjects will be recruited. The first 5 will undergo lymphostem cell treatment, and the subsequent 15 will be randomized in a 2-to-1 ratio into the stem cell treatment or the scleral lens treatment group. This subject will be undergo screening using the clinical grading system, the in vivo imaging, and molecular diagnostic test to determine the degree of their lymphostem cell deficiency. If they meet the inclusion criteria, the biopsy will be taken from the donor eye, grow them in culture, and then transplant it back to the other eye, the diseased eye. The control will be a scleral lens treatment group. You can contact our study coordinator through email or to me if you need additional information, and the trial is registered at the clinicaltrials.gov. I would like to thank my teams who have contributed to the work that I have presented. Sheila Gonzalez is the project manager who developed the xeno-free and feeder-free culturing system. Alan Pascal is our clinical research center administrator who helped us tremendously in the entire project. Alpha Stem Cell Clinic and the Stem Cell Center at UCLA have assisted us on the R&D preparation. Dr. Donald Kong is the director of the GMP facility, and he has helped us greatly on the GMP tech transfer and also the GMP manufacturing of our product. The project has been funded from the infancy by the California Stem Cell Agency, as well as the National Eye Institute, and the other funding agency also contribute to the beginning of the project. Thank you very much for your attention.